Hello.
Hi, Bradley.
Hi, how are you?
Fine, Bradley. How are you?
All right.
Yeah. This week I did a pretty good one. Last week I haven't done anything, but this week it was quite good. Bradley, did you saw that I have run so many times Hugging Face Spaces repos actually 2 days back? They are not working. There is a bug error in them. Suddenly they stopped working. So I tried to implement them again and rewrite the bugs. Some another thing also I did. Hope I will show this weekend.
Okay.
And Jyothi and I are started writing paper actually about this thing only. I mean, segment anything model versus DevoNet, which I have proposed in the model proposal. I started coding of that model also. So that's. I will show you.
That's good.
Morning.
Hi, how are you?
In the one-eyed.
Oh, yeah.
Land of the one-eyed. Or land of the blind. One-eyed is king.
Yeah.
I've got one good eye now.
Oh.
Okay.
Okay.
They do the surgery separately for each eye.
Oh.
So I don't know how long it's going to be.
Oh, okay.
Find out tomorrow, maybe.
Yeah. All right. Yeah. Well, it's too bad to hear.
Well, it's not too bad. The vision's excellent. One eye. Yeah, I just have to cover up the other one.
Yeah. So,
it makes it difficult for a project on stereo vision.
Oh, yeah. So, Himanshu, could you bring up your screen, the Hugging Faces space, so I can see what you mean?
Yes. Could I share my screen and show it to you?
Yeah, go ahead. Hello, Jyothi. Welcome.
Okay.
There we go.
Yeah, 2 days back, the spaces actually stopped working due to some library issues. So I changed everything. I added notebooks also, training notebooks, which was written by Mayuk, and I changed this requirement.txt. And yeah, these are the commits I have kept last week in this repo. Like this, all models have stopped working. Everything, this nucleus segmenter, membrane segmenter. So I made them to rework again, and my work for the last couple of weeks was on segment anything model I have worked. This is the repository. The main problem is I have added this code from segment anything repo to here to import it easily, and the notebooks are here which I was working on right now, and utils here. The main this is a loss function which was which I took from another paper actually to implement. Segment anything model loss function is kind of different. It is implemented in different way. It took the mean values of loss values of from a dice loss and then implemented in JCOD loss, and this is the whole code for it. This took a lot of time to write and understand it and integrate with my model.
Yeah.
And this is the code actually I was working on. Almost it is over. I should just need to add the data set and start training it, and little bit of hyperparametric tuning also needed for this thing. And this is a kind of extra work I did. Bradley, I hope you remember this thing made by Minux, in
Yeah.
last past GSoC. He hosted in Heruko, but Heruko has removed its free services and it couldn't work. And Hugging Face Spaces also, it is not working properly. Sometimes it is stopping, and there is a problem with rerunning them all. So to clear that, I have took a server which was on render. This is the link of it. I hosted them on render. And they are working actually, but they need some, there are little bugs I need to solve. If those bugs are solved, we can keep this working like as fast. And our DevoWorm organization in Hugging Face also keep the we can keep them running. And SAM model will also be ready by next week. I just need to start them training actually. That's the only part left. These are the notebooks. Actually, last week I haven't worked, not past week, before past week I haven't worked that much. So I didn't took that as a first week one of my GSoC period. I took this week as my GSoC week one period, and I'm gonna write the blog about what all work I did this week. This week I mainly implemented this notebook, how to segment, fine tuning the SAM model. After that, I started with in working with Hugging Face spaces to remove the bugs which we are encountering. After that, I hosted all the code into render to segment it.
Yeah.
This is the work I did. Maybe by next week, segment anything model will be ready, and after that, I will start working on the DevoNet model which I have proposed. I need to update. I wrote the code for it, but it is in Python 3.7 version. I need to update it to Python, the latest version of Python, and train it with the C elegans data. After that, we want to create the comparison between segment anything model and DevoNet model and start writing paper on it. Jyothi was actually helping me with the paper. We two are writing. As you are the head of this project, you will be the what we call co-authors of this. You, me, Mayuk, and Jyothi Swaroop for the whole paper. I think this paper could be a pretty good thing because SAM is trained on around 11 billion masks and 11 million images and 1 billion masks. But DevoNet model is just trained on the C elegans data of 6,000 images. If we able to get a good accuracy with the DevoNet model, we can write paper on it, or we can at least make a comparison between them. Like this model is outperforming SAM model. I hope it will outperform the SAM model. So it could be a good thing in cell biology to segment.
Yeah, it sounds great. Yeah, it sounds great. Have any idea why the Hugging Face repository isn't working or wasn't working? I mean, is it like a problem in there on their end?
Not there on their end. Actually, whatever code I have wrote, it stopped working due to those are all Python old versions. Dependencies are deprecated actually.
Okay.
So I need to update those all dependencies in the Hugging Face. When I updated it, it started working actually 2 days back. After that, I hosted this render thing also. I need to make some small changes in this also because when I click here run, it is not working. See, I need to rewrite some things. Yeah, it is working. Yeah, it's working. Sorry, but there are so many deprecated things in here. So yeah, Bradley, I will add this repo after changing some code in it. I will add this repo into the main devolearn, and I will keep this link as the who can visualize them. I can see. Hugging Face will be like another platform who can see our transformer spaces models. This will be like official. Anyone can open and see. This will not stop like Hugging Face spaces in the 2 days after or 3 days after.
Yeah. Did you? I don't know if you got like alerts about deprecated files, but I get them all the time for stuff. So it's kind of like a pain.
Okay, I didn't got anything message about this deprecated libraries. Suddenly, I opened Hugging Face for hosting segment anything next file. When I saw these 3 models are not working, then I started digging out what is the wrong going on. So when I encountered, I saw like these all libraries are deprecated. So I changed a little bit.
Yeah.
We don't have idea to how to start a paper. I mean, we haven't written till now anything in new paper how to write. Do you have any guidelines or some kind of instructions we could work on that? We know to write as abstract at starting, but after that, what structure we need to follow, we don't have an idea about it.
Well, yeah, so the paper is going to be on the method, right, mainly?
Yeah.
So,
segment anything.
yeah.
They want to compare you.
Yeah, so actually the best way to start writing a paper like that is to do an outline. And outline like what the method is, why it's important. So why do we care? And in this case, it would be applying segment anything to biological data. And I can help you with the significance. And then going to the methods, just have step through the methods one at a time. What is it involve? Does it involve, these libraries? What do they do? How are the dependencies? How are they related? And that sometimes you'll put in like a demo or a couple figures to show that what you're talking about in the paper. So oftentimes, I don't know if you've ever seen what's the journal they used to run, distill.pub. They've written some nice interactive papers where they implement, it's basically a coding method for something, but they do it like where they put the interactive demos in the paper, but it's written like a paper.
Okay, understood.
Yeah, so I mean that's the way I'd go about it, and then I'll help you with the drafting of it and everything.
Okay.
Yeah.
Okay, I will create a doc for us, you, Jyothi, me, and Mayuk. Common doc. We will start implementing. Jyothi and me, we will start writing the paper. If you weekly, once you check it out, how it is, just give us some insights, like you did with my proposal, right? I mean, corrections. Yeah, that would be nice.
Yeah, no
that's all my updates are. I will consider this week as my week one, and I will write my blog and explain everything what I did in this week. And these all are my updates. But by next week, I will try to complete segment anything model and start working on DevoNet.
Okay. Yeah, that sounds good. I look forward to it. Yeah, thank you for the update and kind of explaining everything. And yeah, it happens a lot where you get things that are deprecated. And I know GitHub will send alerts, sometimes too many alerts for different things, but that's where sort of the maintenance part comes in. That's why we kind of we have projects every summer. We try to get people in to work on the platform, to improve the platform, but also to maintain it, make sure things are working, because you get a lot of we don't have like a dedicated workforce on the platform. So when you're developing open source software, sometimes you have to just rely on people to come in and maybe work, come in for a while and work on something, and you'll find that there are broken things in the code and things like that. Sometimes things will break just at random if a dependency is hit wrong. So it's always a struggle to keep that up, And so that's a lesson for maintaining open source software, is that oftentimes you have that problem where things get deprecated, things get, come into conflict, and it's just a matter of, patching it up as you go along, because you don't know what's going to be deprecated, in 6 months. It's just kind of a thing that happens. And hopefully enough people notice.
Actually, this is a repo I have kept in the chat. This repo is kind of dependable. We can integrate with our repos. It automatically, it's an AI actually. It automatically clears the deprecated libraries and add new libraries, and we will try to integrate in this thing into DevoLearn if possible. You told me right now you are getting messages from GitHub like deprecated libraries are there. So we will try to integrate. Jyothi has a good experience with this repo. We will try to integrate with our DevoLearn repo, add it to every repo, this library as a package. So it will directly update the deprecated libraries by its own when they are deprecated.
Yeah, that's great. Yeah, it's usually the best way to do it is through bot, through automation. So, and then yeah, I look forward to seeing Jyothi as well. If he, what he can do on that, and we'll work on
Actually,
that
he was
paper.
selected for another GSoC drug project
Okay. Yeah.
under INCF only, but he was parallelly working with me also on the DevoWorm model.
Oh yeah, yeah, that's great. That's wonderful. Yeah, congratulations, Jyothi.
Thank you.
All right. So Himanshu, how are you?
Hi, Brady. I'm fine.
Hi.
I'll start with an update.
Okay.
Let me just share this. Is it visible?
Yes.
Yeah. Okay. So basically, I was working on instance segmentation and trying to do the pre-processing work of a few data sets, and I also came across a new data set on C. elegans, which I found like really interesting.
Okay.
I'll start by showing that first. So basically, this is a 3D nuclear instance segmentation dataset of similar to ours, like the fluorescence microscopy volumes of C. elegans. And it is like a this paper is like an iteration of previous papers, in which the first paper was about the dataset, and in the next paper they did like augmentation on it, like data augmentation, like just before doing the segmentation part. And in this paper, they are basically finished like the entire data set and given like a directory wise, like test train and validation data for it. So I tried to visualize what it was given, like similar to the code which was written last week, and basically the data looks something like this. So it's like a huge line, and it has the entire worm, and that's how it visualizes. Like these are the individual cells. So uh, this was like what I started with, and this is basically the masks for the same cell, like TIF file. So this was like the new data set which I found, and I'll try to run my model on this as well. I just have to resize the images and do a few tweaks. And I also worked also simultaneously uh, on the cell membrane data. Like I did the basic pre-processing of it and kept it ready so that I can use it, and which is in this pre-process segmentation data file. And also I've been working on like a mask CNN model, which is basically like it's similar to something called as a it basically does instant segmentation in a way which is like it's basically a concatenation of two models, you could say. It's like a faster CNN model, and along with that fully connected convolutional neural network, so it's an FCN. So what it does, it does pretty much everything that a faster CNN does, except of the region of interest is like in faster CNN it uses ROI pooling, but in this it uses ROI align, which basically changes the stride values. It's like the internal thing, and due to this we get the object detection part of the code, and after that we use an FCN so to get the segmentation. So this is how instance segmentation is done, and it is one of the state of the art models which has been used for a long time. Also I've kept two backup models ready on which is like on Hugging Faces, and I'll try to implement that if this doesn't work.
Okay.
Right now I'm getting an error in the train my epoch stage. I'll try to find out what it is about. And I also came across a few more papers and interesting stuff which I've just kept it in this file. I'll just share it, whichever I find interesting.
Yeah.
So so hopefully in this week I'll try to complete the instance segmentation model, and in the subsequent weeks, I'll do the topological data analysis and the GNN parts.
All right, that sounds good. So this new data set that you found, could you pull up the Zenodo hub or the Zenodo stub again? Yeah, there it is. 3D nuclei instance segmentation, fluorescence microscopy volumes. So this is 28 volumes of C. elegans L1 stage and corresponding stacks. So this is where the worm is hatched, but it's still like a larva. So it has the anatomy of the worm, basically, from head to tail. It's unfurled now. And then 28 raw images and corresponding masks of average dimension, which is the thing that they define here. Pixel size, it's pretty fine grained. Then microscopy. This is the citation. Yeah, I don't think I don't know. I'm probably familiar with it, but I don't remember right now. Then these are nuclei masks. So for the whole worm, so it gives you some anatomical resolution, I guess. So you know what cells are at the head, what cells are at the tail. So these are the files. What was that?
I'll also get into like the naming convention of it. It's basically like some stages or some part of it like that, which is why it was named this way.
Okay.
Like, yeah.
Yeah, it looks
so
like. Yeah, go ahead.
No, sir. Basically, it's like at an L1 stage, and this is some kind of. It basically tries to tell us something like it's at this stage and at this particular embryo type. I'll just look into it once. Okay.
Yeah. Well, I mean, some of them I think might be I'm judging from the name here. It might be like different samples because it looks like they're like kind of similar stages, but some of them are a little bit different. Like 4L1. I mean, they're different. I think they're different markers, maybe like ELT3, HLH1, and it's just where they pick a certain gene to express and get the fluorescence data out of it. So you have to pick a gene that's expressed maybe in certain cells or in all cells, so you know that it'll fluoresce when you put it under a microscope. And so that they'll do that. Sometimes that's what the first part of this is, and then L1 is the stage in development. So that's the first stage after it hatches, and then the number after, I guess, is the sample number. I don't. Yeah, the paper will probably have all the details on what those mean. So yeah.
It's probably this one because this is just basically a data set, and which was on a Zenodo.
Yeah.
So I'll look into this as well.
Okay.
So, yeah.
And so the plan is to use this as a mask, like for identifying like fluorescent cell types, so you know what the shape is, what it knows what to look for, basically.
Yeah.
So you train it on this data set, and then yeah.
I'll train it on this data set and try to find the instance segmentation of the basically. So it's kind of over here. So we'll find like in the 11th stage how many of the nuclear cells are there, like, and it will try to particularly check if for each nuclear cell how the cells are over here.
Yeah, so it can identify single cells, or because they get the fluorescence signal is usually pretty clean if there isn't a lot around it, but sometimes you get like noise, and it's something that just happens with because you have tissues underneath. Sometimes there's what they call autofluorescence, where you get interactions with the tissue. Sometimes they're very close together, so it's important to make sure that it's training on single cells and not like a clump of what will be interpreted as a clump of cells, because you have this fluorescent signal in one area. So, it looks like it's probably pretty clean. You might go through and make sure that it looks like it's picking up like individual cells when it, goes like just because it's a mask, it needs to be pretty clean as a mask. So, we've done like manual masks in the past, which are not good because you have to work, it's a lot of work. It's good to have a data set where you can like look at some of the, actually have real features. So, I think this is good. So there'll probably be some pre-processing of this though, or at least like, making sure that it's well prepped, and then you can train your model that you're trying this faster RCNN, so as opposed to an RCNN.
Fast RCNN, which basically is a combination of two models, like a faster RCNN and a FCN network.
Okay. And then you're going to go from there, then you can move to the topological data analysis part.
Yeah.
Okay. Yeah, that sounds great. I'm just just walking through the steps. I want to make sure that we're not missing anything. But yeah, that's a good update. Thank you. So yeah, when we bring all this together, we'll have like, and we've had this in the past where we kind of have this collection of different models, because the problem here is that we want to segment cells, but the other problem is that there's not uniformity in cells, in biological cells. You have a lot of different configurations of cells in an embryo or in a developmental phenotype, and sometimes depending on the organism, we usually train it with C. elegans data. We've tried different other species before as well, but this is largely, we use C. elegans data sets. And that has some limitations because C. elegans has a specific developmental progression. It has certain cell types with certain shapes. So, if you want to go, say, to a Drosophila embryo, you may or may not be able to use the software very well to segment out those cells because it's a different process. There's a lot of cellularization. There's a cellularization stage from a syncytium and things like that. So there's a different set of things going on there. But yeah, so it's nice to have like multiple models where we're kind of seeing, we're doing different things, and then we want to be able to put those together. And eventually, maybe, we say that there's a pipeline for getting, one answer. And if that doesn't work, you can try another set of models to get something else. And so we give, like, at the end of the day, we give people options of what they can use, different tools. It's kind of like different types of pool cues or something, when you're playing pool. People like different types of weights and things like that. So it's good to have multiple models for multiple tasks or different specialized tasks. But I also want to make sure we don't replicate any work too. So I think right now we're in pretty good shape though. All right, yeah. Thanks for the updates. Looks like things are coming along pretty well. Looks like Hare Krishna's here. And Susan. I know Susan said she might not be able to make it, but she's here. So thank you for showing up. Hare Krishna, do you have an update for us, or I haven't seen you in a while?
Yeah, hello.
Hi.
Yes, I haven't been here in a while. I was mostly busy with other but recently I was again working on my last year project because I thought the place it was left last year should be continued further because it can be improved further.
Yeah.
So I planned to integrate ML into it using by implementing a NUS model. So I have begun to write the code. I somewhat completed the code. I need to first test it on a data set. So I'll use the last year's images which I got from Susan, and I'll test if it somewhat works. If it's a positive output, then I can use a new data set to get a better output.
Yes, I just sent Bradley more images, and I'm going to put some up on Sync for Richard or Dick for
Thank you.
so everybody can share them.
Yeah.
The second egg turned out fairly well, and so the first egg not so well. So, but I'll just put all of the images up.
Susan, you want me to create a URL so anybody can view it?
Sure. They're
Okay,
to share. I made them to share. So.
Okay, I'll do that.
Okay, I just need to get them there. I've been, I have a large meeting tomorrow with my advisors.
Okay.
So
they're trying to outnumber you.
So if they don't arrive until later in the week, that's what happened.
That's okay.
Yeah, it'd be great if Hari Krishna could get his hands on the new data. So if you could share that, Dick, share the link to Hari Krishna, I think, or to me. Yeah.
I have to first. What I'll do is get it from Susan. I'll make a special folder, and then give you the URL so anybody can read it.
Yeah. Okay.
Okay.
Yeah, that sounds good.
Okay, so that'll take depends on when Susan gets it to me and give me a day.
Yeah.
Probably Wednesday.
Okay, fine.
Oh, yeah.
Send me, Susan. Send me an email message when you've done it. So, because Sync doesn't tell me about changes.
Okay. All right. Yes.
Okay. Thanks.
Yeah. Thank you. Yeah, I did get it. I did get a copy of the data in an email, Susan. So I'll look it over on my end. I just don't really have a space to put it in right now. But yeah, if Hari Krishna, once he gets access to it, yeah.
I've got over a terabyte of space in the cloud.
Okay.
I'm okay.
That's good. And then yeah, so I mean, once Hari Krishna gets the new data set, he can keep working on the thing. But yeah, I mean, it'd be nice to have like a update on that just to see for the new people too, who I know people haven't been here last year. If they could see what it is that you did last year and how you want to improve it, that'd be good. I mean, it doesn't have to be today, of course, but it can be like in the coming weeks. We can go back.
Bradley, what I can do presentation.
Yeah.
Bradley, I can give you editing privileges once I set that up. So if you want to add things, you could do it.
Okay. Yeah.
Okay.
Yeah, that's good. Thanks. All right. So.
Yeah, there's only one thing to keep in mind, and that is I've got over a terabyte, but anybody who wants to use Sync for free is limited to 5 gigabytes.
Oh yeah.
Okay.
Yeah, so you have to limit.
Okay, so while I don't have to pay attention to the total, other people do.
Yeah. Well,
of course, I think they can download selectively, even if the folder itself is over 5 gigabytes.
Oh, so they just download a part of it, and
I think so. I'm not sure. We'll have to test that.
Well, yeah. It's always getting around these limits. That's the.
Yeah.
So, yeah. All right. Susan or Hare Krishna, do you have anything else you want to add?
No, today was busy, and next week is also busy. I don't know if I'll make it Monday next week. It just the way things are here right now.
Yeah. That's okay. Yeah. Well, good luck. Yeah.
Yeah, they want me to finish this project, the tensegrity project. It's like
Right.
yesterday, please.
Yeah.
Anyway, yeah.
Yeah. So good. So I guess we'll move on to some things here. Now I promised that I would talk about this workshop I went to. I think it was 2 weeks ago now. And so I can't share any of the slides or anything, like screenshots, but I can show like the agenda. So I'll do that. Let me share my screen. So this is SynCell 2023. This was a virtual conference. It is hosted in Minneapolis, Minnesota, at the University of Minnesota, but it's like, it was online. It was on Zoom, so access to the lectures. So this is about like building synthetic cells and building the parts of synthetic cells. So why would you want to build a synthetic cell? Well, you want to build a synthetic cell maybe to synthesize biomaterials, or you want to build a synthetic cell for modeling like a disease state or something like that. So the conference was interesting. The keynote speakers kind of got into like why you would build a synthetic cell, some of the recent progress that's been made on it, and then the panels that they had, or the sections, that are not keynotes. Those were more about like different parts of how to do synthetic cells and build them. So it was a really interesting conference. If you've heard of the J. Craig Venter Institute, they're in San Diego in the United States, and they build a lot of these, like genomic tools. J. Craig Venter ran one of the human genome sequencing projects in the 90s. He founded this institute, and they've been working on not only building genomic tools, but building these synthetic cells. So they've made some really interesting progress in this area. So this is kind of from the conference here.
Bradley?
Yeah.
If you have the URL handy, could you put it in the chat, please?
Yeah, I will. Let's see. Yeah, buildacell.org. There we go.
Thank you.
Yeah. So we had, let's see if we can find the program. So they had these sessions, and they were kind of going, they had like people worked on these different areas. They had it broken down into these cell-free translation systems, the cytoplasm of synthetic cells. They had 2 sessions on that and some posters. They had Beyond the Bench, education, outreach, ethics, and biosafety. This was one session of the poster. They had something called containers, which I was really interested in. And this is where you build containers like lipids and what they call coacervates and microfluidics. Microfluidics being systems you can test your containers in. Liposomes being like vesicles that you can pack things into. Coacervates are these sort of aggregates that we've talked about in the meetings. So these are things that have to do with building a container for your cell. There's a lot of stuff that intersected with early life. So we've talked about early life, the origins of life. And, so some of this is like engineering cells for, applications. But some of this was like looking at how life might have started, thinking about like how maybe life started to organize. And so the container sessions were really sort of focused on that intersection. So it's really interesting stuff. There were 5 sessions on containers and then posters. The first session for containers was, they had something in synthetic organelles. They had things about designing synthetic cells to protect from antibiotic resistant bacteria. They've combining models of membranes and intracellular condensates, non-lamellar lipid compartments for artificial cell development. Actually, they had some things on micro robots and other types of automation at the micro scale. So there are things like that as well. So it was a really interesting set of talks in the program in the containers sessions. Then there was building blocks, which is where they looked at metabolism, gene replication, membrane synthesis, and ribosome assembly and things like that. So these are like the building blocks of a cell. Like what kinds of things need to be there for a cell. And again, this is mostly like for building cells for applications, but some of it also was this sort of idea of early life and how you build a cell or some sort of vesicle. And so they had some interesting sessions. So Lynn Rothschild, who works at NASA, talked about extremophiles and how those can aid in building cells. There's this project, the Build a Cell project, where they were looking at building synthetic extremophiles, which are bacterial cells mostly that live in extreme environments, like really hot environments or really oxygen poor environments, things like that.
Are they trying to build extremophiles that are more extreme than the ones that have been found?
I don't think so, but I think it was like one of these student organizations where they were trying to get like a hands-on experience with like synthetic biology. So they were building cells. They were just doing these experiments where they were trying to build different types of extremophiles, and they call them hell cells, which are like they live in hell,
Sounds like the Terminator.
Yeah, Terminator cells. Then there was this artificial cells from soft matter to cell-like behaviors. So there was a soft matter component to this, which is that you have these cells that have behaviors, and they behave according to the principles of soft matter. So you have all sorts of things that you can look at there. You have spatial organization and cell signaling, so you can actually use these models to look at those sorts of questions. They had a lot on like cytoskeleton and actin and things like that, and their role in some of these synthetic cells. So it's all stuff that we're very familiar with from the meetings. And they had an applications section. Say anything useful in money making, like medicine, bioengineering, etc. So basically, if you can commercialize it, that's what that session was. The cytoskeleton session, which was protein, DNA, and other structures. They had some interesting talks on like protein and DNA, like engineering, like structure, secondary structure for DNA and proteins and things like that. So it was interesting stuff there. I didn't really attend any of these, but and then minimal cell on live chassis, which is mycoplasma and others. So this is just kind of putting what they call minimal cell, which is what are the minimal parts that you can put together in a cell, and then having this in a living cell. So the session was let's see if they had.
Did they discuss simulations of the minimal cells?
I'm not sure. These are the posters for minimal cells on live chassis. So, yeah, I don't think they did. They had toolkits for genome transplantation, physiological characteristics of a genome minimized. One of the things they're doing at the Venter Institute is they're building these minimal genomes where they knock out all the genes that aren't necessary. So you just knock out one gene after another, and if it doesn't kill the cell, then it's not essential. And so you just get a map of like all the essential genes that you need, and that's your minimal genome. And then you can put genes into the genome. It's a circular genome for a bacteria. And you can put genes in to see if you can add function to it. So, you can characterize the minimal genome in a minimal cell. You can put in genes and see, for different things like, synthesizing different chemicals and things like that. And then you can do all sorts of other things. You can look at cell, like in this case, they were looking at cell division by getting rid of all the non-essential genes and then adding in new genes. So they were talking about, in this poster, they have this Craig Venter Institute Syn 3.0, which is this model of a synthetic minimal bacterial cell. And this is derived from mycoplasma, so that's why they talked about mycoplasma. And then this genome contains only 473 genes. They're able to use this to sort of study the basics of what's needed to build a cell. And then what they can do then is add in genes to add in different functions like cell division. So for a minimal genome, they don't have cell division. It's just sitting there, maybe with some minimal metabolism and things like that. They can actually take these minimal genomes, start adding in genes that we know are involved with cell division. And we've talked about FITZ in some of the meetings, SEPF, and some other genes. So you add those in, and then you can restore cell division. So that's really interesting that you can get it down to that minimal genome and then expand it out a little bit with things that you've identified from other studies that are involved in a function and actually add that function in. So it's a good way to look at basic biological questions like, how do you get cell division from a very minimal genome? So you add in a couple genes and it works. You can restore cell division, or you can restore some aspect of metabolism. So it's kind of where they were going with that. And then, of course, they did a lot of stuff with cytoskeleton and actin molecules. They did some things on mechanical behavior, positioning of actomyosin rings and vesicle blebbing. So they were able to look at some of these sort of engineering minimal sets of modules instead of just a genome. So now if you want to have self-division in a cell, what kinds of modules do you need in the cell? So do you need things like actin molecules behaving in a certain way? Do you need certain functions like movement, or for cell division you need movements? How do you get those to come online, as it were? So this is really a question of origin of life, but also of development, and also of course engineering cells to do things. But you can actually apply those lessons to some of these other things. So that was that conference. I can't share any of the screenshots or anything because it's secret. But yeah.
Bradley, one way of reducing a minimal cell is to cheat and supplement it with stuff it should be able to make, and then destroy the genes that it needs to make that thing.
Right.
Did they do that? In other words, what did they feed these minimal cells?
They didn't really get into the details of the medium, but I imagine some sort of medium where they, I don't know.
Yeah, I mean, it needs an energy source, but what I'm saying is, if it's got to make thiamine, you can load it with thiamine and then destroy the genes that make thiamine.
Right. Yeah.
Okay. I'm not sure if that fits the definition of a minimal cell.
Well, yeah. I mean, you can add things into a medium and supplement them just to, if you want to do that, if you want to take out like some genes that
Yeah, it might be a route to get a real minimal cell, but when it's highly dependent on a particular medium.
Right.
I should have maybe gone to that meeting if I could have. I have a spare dog today. I need to know where the actin ring is in the where it is depth-wise, because some cells have the actin ring at the bottom and some have it at the top, and I'd like to know which cells have the actin ring where.
Yeah.
Did they mention anything like that?
Yeah, well, they talked about some localization, but I don't think they did like systematic experiments.
Susan, do you get actin rings in individual cells or only in epithelial-like cells?
It's as far as I know, there's actin on the exterior of every cell. Like it tends
But
to
the cells are attached.
The edge of the cell.
Yes, but are the cells attached to each other?
Yeah. It's part of the attachment.
Okay, so you don't know. I think in the paper that Wayne and I did, I can't remember the date. Cell state splitters. We gave examples of individual cells with rings, but I don't remember if they were actin rings or microtubule rings.
Well, I have a paper that shows that actin is on the exterior of the cell. Like it goes
to
the
individual cell. Yes, but is that a cell that's not attached to other cells?
Well, it has to be in attached cells because the actin-myosin attachments are what attach cells together in adherence junctions. So if you've got an adherent junction, it's supposed to be a actin-myosin complex, according to what I know.
Okay.
I'm not a biologist.
Okay, that doesn't quite answer the question, which is, does an isolated cell ever have an actin ring?
Yeah. Apparently.
Apparently, yes. Only for cell division, or is it always there?
It's always there. I don't know. Like, trying to read biology papers on this is like, first off, where's the paper about it? And second off, what are they talking about? Or yeah.
They're okay. You know the old work by Rappaport?
Yeah.
Okay, where you measure the force produced by these rings.
Yeah. Okay. Rappaport. Yeah, that's good.
Yeah, I think it was the starfish eggs.
Okay. Yeah, I know. I have a bunch of his stuff.
Okay.
But I also have a paper about. They look like they're cells, intestinal cells, and the ring is at the bottom.
Okay.
And then they show a ring at the top. I'll find the paper and I'll send it to you.
Both
so yeah, they can be either way. And if it's a soft cell, it's at the top, and if it's a non-elastic cell, it's at the bottom.
Okay.
Soft, hard cell, it's at the bottom. I'll find the paper. I need to read it again. I need to find some more papers besides that one, so I can do proper um, proper simulations. Yes, I have a Bailey with me this morning because he went and followed somebody here, and now he has to stay here with me because he wasn't supposed to be here. Yeah.
Yeah.
Oh, I'll put him over.
So I had one more thing to mention here. This is actually quite interesting. A long time ago, we talked about using the synchrotron for different types of microscopy, and so I'm going to put this in the chat. This is a proposal to use this for expansion microscopy. So this is feasibility of mapping the human brain with expansion X-ray microscopy. The human brain is just one, application, but basically this kind of goes through some of the kind of thinking through how this would be used for doing this sort of imaging. So this is Logan Thrasher Collins. So he does a lot of stuff in biotech, and Jesse actually knows who he is. So now Jesse, I know he's not here today, but he'll appreciate this. So this is synchrotron X-ray microscopy. So synchrotron is actually like a particle accelerator that they use to power the microscopy. This is X-ray microscopy with expansion microscopy. So expansion microscopy is where they put in proteins into the sample and they expand the sample. So it's like, if you have a very small sample, maybe 50 microns in size, a feature that you want to look at, you can either magnify down to that scale and hope you can get a good resolution, or you can put these proteins in the sample and expand the proteins in size, and it blows up the feature size. And so it just makes it bigger so that you can see it. And so that's what they do. It's a relatively new technique. So, but they're combining these two things, and they want to look at whole brain human brain connectomics at the nanoscale. So they want to look at features at the nanoscale, which are going to be too small to see with conventional microscopy. They're coupling the synchrotron X-ray microscopy, which gives resolution, and then the expansion microscopy, which gives additional resolution. So this just works out how this might be done. I thought this was interesting because we've talked about this in the past, and it's very much a proposal. I think there isn't really any any images.
They need some volunteers.
Yeah.
to have their brains fried.
Yeah. Well, I think it's probably like in corpses or something, but yeah, I don't know how they do. I don't know how they're going to do this, but so yeah, so it kind of works out some of the technical details. In this perspective, I will explore balances between synchrotron optical engineering choices and expansion factor. So these are all technical details. Propose methods to successfully implement this in the context of human brains and estimate how much it would cost to image the human brain in this way. So this kind of goes through. I think they should have started with maybe C. elegans or something, because
what do they expect to get out of this?
What kinds of images?
Yeah, well, I mean, what kind of information?
Well, you have let's see. So I guess there are things that are very small in the human brain, like very small structures, like synaptic structures, or other types of extracellular matrix, and getting a really good set of images that show those things and how they interact. So we have a lot of really small things in the connectome, different different ion channels and synapses and things like that, and to get a really good resolution of those things is important, I guess. But again, you'd have to target these structures, and you have to really kind of get into the tissue. So having a good sense of what's there already is important. So you have to register the images with what you know. So
okay, is Is this in the intact brain or in tissue slices?
I think it's tissue slices. So yeah.
Okay.
So he talks about that this is a promising alternative to electron microscopy, which is another method that they can use to look at very small structures. And then this modality also falls short when considering the volume of the human brain, particularly after expansion. So, they're doing this expansion, they're doing the microscopy. They utilized 8-fold expansion and lattice light sheet microscopy on Drosophila central complex, which is a part of the Drosophila brain. They used 3 colors to visualize things into voxels of 30 by 30 by 100 nanometers. So that's a pretty small voxel area. It's just the area in which each part of the acquisition you have. So it's a pretty small resolution. But even accounting for the 8-fold expansion, this amounts to a volume of less than 0.5 millimeters cubed. So these are very small volumes. If you expand it, further, you can get a bigger volume, but it's still extremely small feature sizes.
Oh, okay. So they're not planning to digitize the whole brain and figure out how it works.
No, I think they want to get specific places in the brain, specific locations, and see. Yeah. Yeah, because it would take he said, in this example of the Drosophila central complex, getting this kind of data, if you you did the whole brain, I think it was in mouse brain, this estimate is, you would take 876 years to acquire the entire brain.
Oh, there you go.
So, it's not for the whole brain, but it's...
I used to be a member of a group that was interested in brain upload.
Oh, yeah.
Okay.
So, yeah, this is... And then...
I don't have enough storage for that.
Oh, yeah. So this is...
There's a science fiction book about brain uploading. It's called Bob, or the Bobverse.
Oh.
Bob. So you can look that up if you're interested.
Yeah, if Bob didn't live the 800 years, needed to upload his brain. Huh?
Yeah.
Oh, well, they uploaded his brain and made him into a kill sat. Anyway.
Okay.
A probe.
Yeah.
In outer space.
So this is
It's actually a good book.
Oh, yeah.
Okay. Bradley, have you tried getting AI to get the Google money instead of students?
No. So this is a synchrotron. This is a particle accelerator. They have. This is just what it looks like. It's a big building. You have these. You have to have a lot of infrastructure, but it's all particle physics in here going on. So you're generating electric and magnetic fields, and you're applying this to this problem, and then this is the actual imaging device where you have a sample out of the brain. You're doing, I guess, slices. He puts a brain on here, but that's not really. I don't think you're going to get it out of just a brain sitting there. You'd have to have and then you have the insertion device. That's the X-ray beam being generated by all this. It comes through. It has to be charged up. It's super concentrated, and then it goes through the insertion device to this monochromator, through the sample, and then you acquire the image, and it's then that's your image. And so that's
not quite. Not quite. You have to do computed tomography because the object usually has some thickness.
Right. Yeah. So you have to do some processing.
Yeah, Susan might have been here before.
Yeah. Yeah, he mentions tomography in the last part here. So then he recommends some methodologies. And, one of the hurdles that you have for this method is attaining sufficient feature contrast for capturing clear images. So sometimes you have to clarify your tissue, so you have a lot of proteins and other types of things that are, sort of in the extracellular matrix, kind of getting that removed so you can actually see the structures you want because they're so small. So getting that feature contrast is really important. That's where you get like not only, sort of inference of different things like, but you also have to have like all sorts. Well, you could apply a lot of the image processing technologies we've been talking about in the meeting to this problem. Finding the features and kind of extracting them from a lot of noise in the image. So, so they have this other method called unclearing microscopy, where they actually use chemical means to like clear the sample, and then you can take images of it. So that helps too. But having some combination of that. So this is, of course, the expansion part where they expand. So this is a brain. They expand the tissue, then they stabilize it. They clear the tissue of all the proteins. And again, this is not a living brain. This is a dead brain, but they have to, the idea is they're expanding the features out in space. So they're expanding the size of everything, and then they're clearing the tissue, and then they're stabilizing it and imaging it. So you get like this is a motor neuron here, or a stellate neuron, I guess. And then you can expand it, you stabilize it, then you can image things like the synapses, things like different ion channels on the cell and so forth. You can really get really good resolution on that. So how fast can synchrotron image expanded brains? We talked about that. It's going to be a slow process if you want to do the whole brain, but we're really interested in these very small features. So this is, it calculates out some limits for the number of projections needed to completely reconstruct a single tomogram. You can calculate that. For an image with n equals 1,000, the tomogram will require up to 3,142 projected images. If each projection takes 1 millisecond to acquire, this means that one could potentially obtain such a tomogram in just over 3 seconds. So one should realize that larger tomograms will overall increase imaging speed, despite necessitating more projections, since the tomogram size scales volumetrically and the projection number only scales linearly. So there's a lot of stuff you need to do to acquire like a volume. It takes time, and so there are a lot of technical issues to overcome, aside from just the expansion part and like kind of getting the clearing the tissue. So it offers up some a lot of technical details here. The cost is pretty high, but that's something that always comes with imaging. Okay, so yeah, Dick had to leave. Looks like I think he told me he had a session to attend. So, and Himanshu had to leave as well. So very good. Hare Krishna as well. That's fine. Thank you for attending, and thank you for attending. People who are left. Do we have any comments before we go? Questions?
Nothing badly. Maybe by next week I will start writing the blog. For this week's blog, I will write and I will post it in DevoLearn channel. Yeah, that's it. Those are my updates. Maybe we will start. I will share the doc with you, which we want to write the paper and all stuff. Anyhow, that's only updates are there.
All right. Sounds good. All right. Thank you. Have a good week. Now I'd like to follow up on a few things from the last meeting, reading-wise. So the first thing I want to follow up is the discussion we had on active matter and artificial life. So the first thing I want to talk about is the source paper for the Gray-Scott model. So the Gray-Scott model, as you'll recall, was an alternate form of mathematical model for morphogenesis. You can compare the output with the Turing model of morphogenesis. And the mathematics are a little bit different, but they basically both use a couple of differential equations to produce an output. So the Gray-Scott model was first proposed in the Journal of Physical Chemistry in 1985. And the title of the paper was Sustained Oscillations and Other Exotic Patterns of Behavior in isothermal reactions. So this is actually using the chemical reaction kinetics and looking at that and looking for things like sustained oscillations and other patterns. So last time we talked about the Gray-Scott model in the context of microorganisms using their flagellum to produce waves through a medium, and those waves interacting and producing patterns. There are other things like the Belousov-Zhabotinsky reaction or the Bezier reaction, which occurs during a set of chemical reactions and produces highly ordered patterns such as spiral waves and other types of things. So this is the first paper that proposed the Gray-Scott model. This kind of walks through the model. This talks about chemical reactions in open systems. So we're interested in biology and in natural systems in open thermodynamic systems. So these are things like streams and fires, living organisms, and single cells. Related to that is this concept of a dissipative structure or a dissipative system, which is where you have an open system and the spare energy or the products of the energetic flow produce patterns. So you can think of a flowing river where you have the riverbed, which is the structure that results from the flow of the river or the flow of energy through that system. And so on that structure, then you can have things like trees and other types of things. And so you have structure where, as before, you didn't have any. They also talk about autocatalysis as isothermal feedback. So the feature common to nearly all isothermal oscillatory reactions is autocatalysis. And so autocatalysis is shared not only by the Belousov-Zhabotinsky reaction, which we talked about just a couple minutes ago, a wide range of halide-based oscillators, and the arsenite plus iodate reaction. Those are three things that sort of characterize autocatalysis, but also you see autocatalysis with numerous enzyme systems and chain branching reactions in the gas phase. So these are all systems that are chemical in nature that exhibit autocatalysis and then not just produce reactions, but they produce structure, they produce pattern formation. And then they talk about the brusellator and the oraganator, which is from the University of Oregon. The brusellator is another version of that. Those are complicated chemical self-organization schemes. And so they kind of get into that. They talk about, so the equations that they propose here, which we talked about last week, are based on the stoichiometry of the system. So you can look at the stoichiometry and look at how things are added to the system and produce reactions. So this is kind of how they walk through this. They walk through the mathematics, then they walk through the differential equations, and then these equations can be coupled to produce this morphogenetic system. So there's a lot of math in this paper, and this is sort of the original math of the model. And so then they walk through some results here in terms of autocatalysis. So you see cubic autocatalysis with a catalyst of finite lifetime, non-zero inflow of B. So they're looking at different stoichiometries. They're looking at different flows of energy, and then they're producing this pattern of autocatalysis, which is the pattern formation. The second paper is this generative complexity of the Gray-Scott model. This is by Andrew Adamatsky, who's done some really interesting things in the artificial life community. So this is from 2018. It's a more recent paper. And this extends the Gray-Scott reaction diffusion system to a model of generative complexity. So the abstract reads, in the Gray-Scott reaction diffusion system, one reactant is constantly fed into the system. Another reactant is reproduced by consuming the supplied reactant and also converted to an inert product. The rate of feeding one reactant into the system and the rate of removing another reactant from the system determine configurations of a concentration profile or concentration profiles. And those are stripes, spots, and waves. So you basically have these reactions. There's a stoichiometry, which means that there's a certain amount of that reactant. And then given those parameters, we can get this sort of profile, which is almost like a distribution or an ensemble of things. And those ensembles are stripes, spots, and waves. So that's something to think about in terms of information theory and thermodynamic entropy. And so, we can use the mathematics of those phenomena to characterize some of these patterns. As it stands, we don't have a really good method for characterizing collective behavior other than sort of the qualitative method of that looks really interesting. So, to be able to take what we're generating from these models, and of course we talked about cellular automata last week as well, being able to implement these types of patterns in cellular automata and then analyzing those cellular automata in terms of the different classes of complexity they produce. Now, Stephen Wolfram's work on A New Kind of Science sort of goes in that direction, where he classifies different patterns, classifies the rules, and gives us a good sort of typology of complexity. But there are other ways we can analyze this too, and one of the ways is to sort of quantify different variations in some of these outputs. So, striping patterns. What happens when you change a set of parameters upstream? Do we get a quantitative difference in striping or in spotting or in wave patterns? And what are those parameters? There might be a orientation parameter in waves, for example, or an attenuation parameter in wave formation or in stripe formation, frequency information that we can use to characterize different types of outputs that commonly occur. Because that's an aside from this paper. We calculate the generative complexity, a morphological complexity of concentration profiles, grown from a pointwise distribution of the medium of the Gray-Scott system for a range of the feeding and removal rates. The morphological complexity is evaluated using Shannon entropy, Simpson diversity, approximation of Lempel-Zib complexity, and expressivity, which is Shannon entropy divided by space filling. So I didn't realize that they were going to get into this when I started the abstract. I just mentioned that we should use something like Shannon entropy or the mathematics of thermodynamic entropy, and indeed we get this Shannon and Simpson diversity approach, or Shannon entropy and Simpson diversity approach. Shannon entropy being the information content of something, the H value. Simpson diversity being very similar, but also having allowing you to look at the diversity or the unpredictability of things. So there are different measures that they're using here to characterize some of this morphological complexity. But still, I think my point stands about being able to characterize some of these outputs quantitatively and classify them. It's kind of hard to do, and there isn't a lot of work on this. We analyze behavior of the systems at the highest values of the generative morphological complexity and show that the Gray-Scott systems expressing the highest levels of the complexity are composed of the wave fragments, similar to wave fragments in some subexcitable media, like the BZ reaction, as we mentioned earlier, that allow you to generate these kind of things and these kind of patterns. And so there's subexcitable media, which is where it's not as excitable. So excitable means it produces outputs that are always changing. There are a number of neural models for this. There are a number of chemical models for this. So you get these kind of phenomena. You get subexcitable media, excitable media, and then you get these different patterns that result. Then he brings in, ties in this idea of Conway's game of life and dissipative solitons and gliders, which we talked about in terms of the game of life last time, also in terms of the Gray-Scott model and what people are doing with cellular automata, and then with the sort of linear models that we talked about last week. So that's this paper. Then there's this game of life and the epigenetic principle. This is in Frontiers in Cellular and Infection Microbiology. And this paper uses cellular automata to explore Conway's game of life and epigenetic principles, or what they call epigenetic principles. So we'll go through the abstract here once again. Cellular automaton and computer simulation games are widely used as heuristic devices in biology to explore implications and consequences of specific theories. Conway's Game of Life itself has been widely used for this purpose. This game was designed to explore the evolution of ecological communities. We apply it to other biological processes, including symbiopoiesis. Symbiopoiesis is autopoiesis, but it's also autopoiesis between different systems. We show that Conway's organization of rules reflects the epigenetic principle that genetic action and developmental processes are inseparable dimensions of a single biological system, analogous to the integration processes in symbiopoiesis. We look for similarities and differences between two epigenetic models by Turing and Edelman. So Turing proposed an epigenetic model. In addition, it's different than the reaction diffusion model. And Edelman also proposed an epigenetic model. And so these models are realized in Game of Life objects. So remember, the Game of Life is a cellular automaton, and it has these different patterns that form as a cellular automata does, but it usually is something that's moving. So you have these things like gliders, glider guns, different patterns of activated cells that often move. They either move stationary in a stationary manner, or they move across the board, the cellular automata grid. And so you get these patterns that change, and people have tried to classify them in different ways, and they result from the zero-sum game, or the zero-player game. And so the Game of Life, they implement these models in the Game of Life. They show the value of computer simulations to experiment with and propose generalizations of broader scope with novel testable predictions. We use the game to explore issues in symbiopoiesis and evo-devo, which is the evolution of development, where we explore fractal hypothesis, that self-similarity exists at different levels. So you have cells, organisms, and ecological communities. There's this complexity of each, and that complexity is self-similar. So you have similar patterns of cells, similar patterns of organisms, and similar patterns of ecological communities. So you get these, sets of interactions that occur at different scales, and this may explain why maybe you see, like in single cell communities, certain patterns that get recapitulated to the level of organisms, complex organisms interacting, and then ecological communities and the way they're organized. This similarity is the result of homologous interactions of two as processes modeled in the Game of Life. So these are pairwise processes modeled on the cellular automata. One thing they mention here is Conway made connections with biology, part of his purpose, bringing out analogies with the rise, fall, and alternations of a society of living organisms. So this is from Gardner. So Conway was interested in biology as well as sort of this game theoretic aspect. And so this purpose explains the interest of this game for biologists, since it explicitly aims to model a basic process in biology, the evolution of ecological communities. But you can also apply it to other parts of biology. You can apply it to development, to early life, to general biological complexity. And so this has become a main thing in looking at biological chaos, a main tool by which we can look at these things. So he also talks about some of these distinct emergent forms. So there's still life configurations, which are stable over many iterations. And he shows an example of still life here, where these are things that form and that remain stable. They can remain in their fixed form. So they don't really move around, but they also don't disappear. They're not itinerant. Then there are these blinkers, which are patterns that oscillate with a fixed period. So these are things that are also stable, but they're not stationary. They are, moving around, I guess, in a sense. They're turning on and off. And so it's this blinker where it's an oscillation that can be measured. It's a regular pattern over time. Oscillators are stable over a cycle, returning to an initial state. Such cycles can be short blinkers or very long So you can have blinkers or longer oscillations. And so then there's this other set of things called movers and gliders, which move across the grid. These are emergent forms, so they move. They're itinerant in terms of their presence. You can see them and then they disappear. So a glider might glide across the cellular automata grid and disappear. But these are the things that have really captured the attention of biologists since they look biological. But they're not, of course, not quantified as biological. So, like I said, from the last paper, we have these quantitative tools, but we still need more quantitative tools, I think, more rigor in terms of characterizing these patterns. There's a lot of rigor building in terms of describing how these patterns are built, but not a lot of rigor in terms of analyzing these patterns and the emergence that produces them. So Conway uses a lot of concepts from ecology, and so this leads to their paper where they talk about these ecological relationships. So Turing has a model of development, and it's related to his model of morphogenesis, reaction diffusion morphogenesis. It isn't the same thing, but it has a number of different features. So his concept of morphogens, which are these particles or molecules that he proposes are theoretical constructs, so they can be anything in nature that does this sort of thing. But they basically, morphogens are form producers. They're these fundamental particles of his reaction diffusion model. You have gradients of morphogens, you have patterns that form because of the interaction of morphogens. And Waddington had a similar concept called of evocators. So Waddington's evocators are hormones or even pigments. Again, this is a theoretical term, so it can fit a lot of things into that box. Turing also assumed that the function of genes are purely catalytic, that they persist unchanged after producing chains of effects. So you can have things that are, genes will sit there and produce a number of different reactions, but they can be stored as unchanged things. So in the game of life, there's a very analogous thing where you have this common basic pattern, which is maybe defined as a blinker, and it can be understood as involving catalysts. So the blinker begins with a row of 3 cells following the rules, and the first iteration of the two of the 3 cells dies on either side of the central cell, but 2 new cells grow above and beyond the central cell. So you basically have this template for interaction. You have this blinker where parts of it turn off, new parts are added. You have this sort of kernel of a blinker that has, the cells around it have a frequency, and it basically is recognized as a pattern. But so you could think of this as like having a genetics to it where it turns on a central cell, and that's persistent, and then the cells around it are variable in terms of their pattern, and that pattern is driven over time, so the interactions over time give you this frequency of the neighboring cells. And so you could analyze that as a common basic pattern, a set of blinkers with a frequency, and that may be different over time. It may be very common at that frequency and in that shape. So this is why we want to be able to analyze these a little bit better. Turing also examines a simplified system with only 2 morphogens, X and Y. And so you have like you saw with the Gray-Scott model, you have 2 parameters, X and Y. Turing also looks at the more complex case of 3 or more morphogens. The X, having the 2 morphogen system, the pairwise interactions, the 2 parameters, you can actually get nice striping patterns from that. But a 3 or more morphogens is a more complex system. And so this is something that people haven't really explored too much, that you have, multiple sets of interactions all at one time. So this is actually kind of what we were talking about with some of these patterns in the game of life. And we can maybe replicate some of these. It's hard to say. So one of the things I want to point out about Turing's model I find interesting is that we have this sort of X and Y. So we have X, We have Y, and say this is like a bunch of morphogens in a concentration gradient. So we have this morphogen, these morphogen patches. They'll diffuse in either direction. So they'll start to overlap. So X is moving in this direction, Y is moving in this direction. This region of overlap is where you get the pattern. So you get these nice striping patterns from this pairwise interaction of X and Y. So X is here, Y is here, X is here, Y is here. They diffuse into one another. They form these regions of interaction. And these regions of interaction, instead of being gradated, there should be a gradation, like basically a white stripe, a black stripe, and then a gray interstitial in between. But that's actually not what you see. You usually see a very sharp boundary between the black and the white. Let's say that X equals white, Y equals black, and you would expect from those two to see a gray interstitial. It kind of grades from different shades of gray, but you don't actually see that. You see a sharp boundary that is, so the idea is that these gradients kind of intersect and interact to form this boundary, and it gets reinforced over sets of interactions. Now, let's say we had 3 or more. So let's take like a case of where we might have 4. So we have X, Y, M, and N. And X would be here, Y would be here, M would be here, N would be here. And we move out from there. And we get these concentric circles that have the same pattern of interactions. So as we had before with the overlap, we have the same set of overlaps. And you could draw a Venn diagram to show these overlaps and the sort of the exclusivity of each. So, X and M might overlap, X, Y, and M might overlap. There might be a regional overlap that we can define as being unique. Now, what's interesting about this system is it's not just a pairwise interaction, it's a 4-wise interaction. And so 4-wise interaction is going to behave quite differently than a pairwise interaction. For one thing, when you have a pair like X and M, you'll have this sort of defined boundary. Where you have a boundary between X and Y, you'll have a defined boundary. X, Y, and M, it's less clear what that boundary looks like. So it might be kind of jagged, it might be itinerant, it might move around. And what we can do with that 4-wise relationship is we can actually put it on a cellular automata. We can model it a little bit differently. We can have cells, a cell here and its neighboring cells in a neighborhood. And then from that neighborhood, we can basically modify the rules, the interaction rules. So X and Y, there's an interaction rule here between this cell and this cell. X and M, there's an interaction between this cell and this cell. And you notice that M and Y don't really come into contact. They're sort of catty-corner from one another. So they're distinct, but they're in the same neighborhood, and they share a connection with X. So what that means is that they're part of the same pattern, but they don't necessarily have a boundary, or when they do have a boundary, it's not clear what the rules should be to mediate it. So there are all these interactions where, in time, where things get turned on and off. So these 2 cells might get turned on at t equals 2 to 4. These 2 might get turned on at t equals 4 to 6. And this might recur at like 6 to 8, and so on and so forth. I didn't get 8 in there. But the point is that there's a lot of sort of complex pattern formation, especially over time. And so these sorts of things, these things that are greater than pairwise comparisons drive a lot of this. We can also model this as a set of patterns in a cellular automata. So one last thing I want to talk about with this paper is Edelman's work on topobiology. He wrote a book I think in the late 80s on topobiology, and I don't know how much people pay attention to it anymore. But basically, there's this topo aspect, which are these spatial relationships and the importance of place. So if we go back to our cellular automata, there is the spatial aspect. It's very explicit. We have a spatial aspect in this model too. So these variables are not just floating around nowhere. They're grounded in space. And in fact, the spatial interactions in these concentric circles, or even back here in these gradients, these linear gradients, actually play a big role in what the interactions look like, the striping patterns, the variation over time and space. So we have this aspect of topo, which is what Edelman defines as the importance of place in other spatial relationships, including the Newtonian and mechanical processes envisaged by Turing. Edelman proposed 5 primary processes involved in development, 3 of which he called driving processes, and 2 he called regulatory processes. So these correspond loosely to the distinction between genetic and epigenetic processes. So he defines epigenetics. Genetics, of course, being DNA. Epigenetics are modifications of that DNA or the products of that DNA. But in Edelman's theory, they are all epigenetic. So the driving processes are milieu dependent. So milieu dependence means it's dependent on the environment or the sort of the environmental state. And so that is sort of a different definition of epigenetic, but that's how Edelman kind of deals with it. If you reframe these processes in terms of the game of life, there's cell division, cell death, cell movement, cell adhesion, and differentiation and induction. And all of these can be formulated in the game of life. They're consistent with Edelman's model. They're also consistent with Turing's model and with this game of life, implementable in the game of life. So I'm not going to talk anymore about that paper. You can explore it on your own. So I'm going to move on to another topic that we talked about last week, and that is swarm intelligence and networks and sort of these swarm intelligence on networks. So I mentioned that we talk about cellular automata. We have these cellular automata grids, and partially these things can be sort of discrete versions of these gradient systems. So you can have these discrete versions of the gradient systems where the 2 parameters interact in their gradients. You can basically have rules between cells that manage those interactions. Now, let's suppose that we want to put those in a network. So instead of having a grid like this, where we have individual cells, we have a network, which is discrete. And again, discrete, but it also isn't really, it can be spatially distinct, but it doesn't have to be. But it basically has these nodes that interact, and their interaction strength determines their connectivity. And it's a different way of describing this cellular automata, except that the cells don't live or die. They're either connected or not connected. So you have different topologies that form. You have different shapes like cyclical complexes, and you can do like all sorts of topological analysis. You can do connectivity analysis, so you can say if something is highly connected or not highly connected. You can do all sorts of interesting things with that. Now we talked about in one case where networks might have this sort of collective behavior. And so imagine that these networks, whatever their topology, have a collective behavior inherent in their topology. In other words, on their surface, you have like nodes that will behave in a certain way together. And in some cases, that's what a neural network is. It's basically if you're connected, if you're highly connected, if there's a strong affinity for connection, you behave together as opposed to apart. So that's where we get into the swarm intelligence. So there was a mention of this in the meeting, and we didn't really know where to find references on this. I thought, yeah, I think people were doing this in like communication networks, and indeed that's where the literature is. So there are 3 papers. Well, this is an article from LinkedIn, believe it or not. This is on swarm intelligence, helping with network optimization and dynamic and complex environments. And this is more of like about engineering. So this is where we want to optimize the network topology. We want to improve performance, efficiency, and reliability. So this is mainly for communication networks like cell phone networks or other types of transportation networks. But so people are interested in this. And of course, swarm intelligence is something that we find in nature. We find this amongst collectively behaving organisms, such as fishes, bees, birds, and ants. We talked about this last week as well. There's this paradigm called swarm intelligence, which is a programming paradigm that takes inspiration from this and builds like these swarms that can be evaluated for their sort of efficiency and their robustness and stability. So, like, we go back to the game of life where we have these patterns that are stable, some that are, itinerant or fleeting, and we can reinterpret this in the language of swarm intelligence and swarm intelligence on networks. So these are collective behaviors that operate on a discrete network. And so we can use swarm intelligence algorithms to sort of evaluate not only the efficiency of networks, but other things such as the robustness of the networks, the other attributes of the networks that may not be immediately apparent just by using a regular optimization algorithm. So they talk about ACO, PSO, which is particle swarm optimization, BCO, which is bee colony optimization. So there are a lot of different methods to these kind of algorithms, and they're all inspired by natural processes. So they talk a little bit more about network optimization, how swarm intelligence can improve. It can allow for people to build networks that adjust to changing network conditions, demands, and constraints, enable cooperation between human operators and algorithms, result in better coordination, communication, and collaboration. So that's what swarm intelligence does in a lot of these communication networks. We can apply algorithmic approaches to this. We can also use swarm intelligence to look at routing in communication networks. This is a paper on this concept. So you're applying a swarm intelligence algorithm to improve routing in these networks. And this is in a communication network where you want to route messages as quickly as possible. And so a lot of these networks are ad hoc. They come into existence, and you want to improve the performance on these networks. So this is a perfect sort of paradigm for this. You have these itinerant network topologies that you want to optimize for on the fly. And certainly swarm intelligence gives you that. So this is, again, from the communications literature. There's a third one here, the Swarm Intelligence for Next Generation Networks. This is a review from the Journal of Network and Computer Applications. And they talk about these next generation networks with communication devices being networked and using swarm intelligence, and as well as game theory and convex optimization to deal with some of these problems. So this is really interesting work. This is the literature where it is now. It doesn't mean that you can't apply these to other problems, other sets of problems, and even back into computational biology and biology. So this is something that's an active area, and I wanted to follow up on a lot of that work that we talked about last week. So thank you for paying attention, and I hope you learned something.